So at the last IPSN Global Partners Forum in Bangkok, we introduced the first investment portfolio. This investment portfolio is supported by the generous contributions of the Rockefeller Foundation, the Gates Foundation, and the Wellcome Trust, but we have added additional funders to the next round of this call, the next round of this fund. And the 10 projects are from 10 different countries across 5 different regions, covering different pathogens as well, and a broad variety of topics within pathogen genomic surveillance. I hope that during the poster sessions, you had a chance to catch up with some of the grantees that are here and that presented their projects as posters. But today we will hear from, they changed them, sorry. Today we will hear from 3 grantees that will present on these are projects that are closer to completion. So without further ado, I will introduce the first speaker, who is Gary Wong. Gary is the head of the virology department at the Institut Pasteur de Laos, and he will be speaking on the environmental genomic surveillance of avian influenza A viruses in high-risk live bird markets in Laos. Gary, welcome to the stage.
Good afternoon, everyone. It's a big honor for me to be here to be able to present some of our work done in the last year on avian flu. So we'll get right into it. Avian flu, just for those of you who don't work with viruses, they can cause severe disease in birds and humans, respiratory disease. The case fatality rate for some infections with some subtypes of avian flu can be up to 70%. So bird flu can be carried by wild birds or domestic poultry, and typically we can classify them as HX and Y. So you may have heard about H5N1, H7N9, H9N2, and so on and so forth. And most countries in the world, so if you look at continents, you look at North America, Africa, Europe, Asia, and South Australia have reported an outbreak, at least one outbreak of bird flu in their bird populations. So in Southeast Asia, if you're local in Southeast Asia, typically if you want to source chicken to eat, you will go to what we call live bird markets, so LBMs. And this is a very common way to buy and consume live fresh poultry. Many of the species of these poultry, geese, ducks, and chickens, are sold in a confined area. And these birds may carry avian flu, such as H5N1, H9N2. And so because when we go to these markets, so whether you're a vendor or a customer, you may have close contact with these infected poultry, and you may, and the virus may spread to you via this mechanism. And actually, in Southeast Asia, so if we look at Cambodia and Vietnam, which are 2 neighboring countries, many cases of these influenza virus infections have come through direct contact with poultry. So this is why it's important to monitor what bird flu is present and circulating in these private markets so that we can detect them before they spread to humans. And so with the support of IPSN, since November of last year, we implemented a pilot genomic surveillance program in these live bird markets. So we chose 2 locations. So one is if you look at the 2 red dots on the right-hand side, in northern Laos, we have Luang Prabang, and in central Laos, so the bottom dot, we have Vientiane. So we chose a live bird market in these 2 cities. And what we do is we go every month and we collect 60 samples, so 20 animal samples and 32 environmental samples at each site. And the environmental sampling can include air, wastewater, slaughtering equipment, so on and so forth, feces. And animals are just direct swabs, oral and chlorical. And for samples that are AIV positive, we try to generate whole genome sequences using nanopore technology. And then once we have the full-length genome sequences, we wanted to see the prevalence, like where the hotspots for AIV are in these markets, and what kind of mutations these sequences have that may make it more dangerous or transmissible or et cetera to humans. So why do we collect different samples? So as you can see, we do direct swabs, we swab the environment, and we also more recently started doing air sampling. And the reason is because we want to minimize the chance of not detecting AIVs in these lab markets. Because we swabbed animals directly, we can see whether we can get co-infected birds, which is interesting, but we may miss some infected poultry because we don't swab every bird in the market. And so the environmental sampling can fill in those gaps, but the weakness of that is that we don't know, we cannot really tell co-infection. So anyway, that's why we do both techniques, and these sample collection techniques are complementary to each other. And so as part of capacity building, we also have supervisory trips and activities with local partners. So these are just two of the more recent trips in May in Luang Prabang and in Vientiane capital. And so once the samples are collected and they come to the laboratory, we were able to put in a brand new platform on how to process these samples and to analyze the data that were generated. And as you can see, with all the activities in red here are new, supported by IPSN. Our current capacity, we knew how to do RNA extractions, and we knew how to do PCR for other viruses. It was not very hard to adapt to avian influenza protocols. But as you can see, after we get a positive result, we were able to put in platforms to that either directly sequence these samples with low CT values, or if they have high CT values, we were able to amplify the viruses in embryonated eggs before we directly sequence them. So this is quite interesting for us and quite exciting. So just a quick summary of the results. We collected over 1,000 samples from the live bird markets since last year, and about 38% of them were found to be AIV positive by screening. We selected representative samples that were positive, and we generated over 40 full-length genome sequences directly from the sample, and also a further 40 sequences from the amplified viruses from embryonated eggs. If we were to look at this data by month, you'll see that we are able to detect avian flu every month, in these two locations. But you may also notice that to the right-hand side, so starting at month 7, month 8, and month 9, there was a dip in the avian influenza virus prevalence in the market, and this is due to and these are because of the months after Lao New Year. So this is where they clear out all the chicken because of the festival, and then they get a new batch of poultry in. We suspect that's the case. We don't really have any evidence to support that even more, but we've seen it in other countries as well, where after major festival, the incidence of AIV drops. And if we were to analyze our results by sample type, we were twice as likely to get AIV positive sample in the environment as compared to animals, so 21% versus 41.2%. But where we were really finding the highest proportion of AIVs was in the wastewater. So this is where they wash the chickens, where they kill the chickens, and also the slaughtering equipment, which is nearby. So we were getting above 50% in these sample types for positive rates. And if we were to subtype them, we mostly got H5 subtype viruses, so about 2/3 of our results of our AIV positive samples were from H5. About 10% of them were H9. We also have about 15% that we were not able to subtype. We have done other PCRs as well against other subtypes, but they did not come back with positive results. And then we have some co-infected samples as well, both from the environment and from the animal, although they were not in very, very high amounts. And if we were to look at mutation analysis for H5 subtype viruses, I'm not going to go through all of them that we found. I'm just going to focus on the one for PB2, so in red. So it's an E627K mutation in the PB2 gene, where we know that if this mutation exists, it's much more virulent in mammals, and especially in humans. And actually, for human infections, especially in Cambodia and in Vietnam that were reported recently, all of them have this E627K mutation. So this is something that we look for. And we also found this mutation, although not in high amounts, but we did find them in our samples as well. So 3 out of 38 sequences. And if we looked at H9N2, we were also looking at similar mutations. But for H9N2, we did not find the E627K mutation. And then if we were to do the phylogenetic analysis of these sequences, most of the H5 that we found belong to clade 2321E, and this is the same as what they found in birds and humans of Cambodia and Vietnam. We also have a minority number of sequences that belong to 2344B, which is the dominant clade worldwide. And the phylogenetic analysis for H9 showed that these viruses belong to clade B471, which is not surprising because it's quite consistent with the sequences from past studies in Laos, Cambodia, and Vietnam. And if you remember at the beginning, I talked I said that we did a little bit of air sampling recently. So this is some preliminary results. We collected 6 samples so far, and 2 of them were positive. However, we found that at the moment, this is very dependent on the surrounding environment. If the surrounding environment is positive, then we were likely to get positive samples, and if it was negative, then we wouldn't get anything. We have 1 sample that is of lower CT value that we can try to directly sequence, and 1 that we need to amplify in eggs, so we're doing that work right now. So we also, earlier this month, held a workshop supported by IPSN to transfer a lot of this skills that we learned from this project to local Lao national partners. So supported by Dr. Eric and Jay at IP Cambodia and Victoria from Wipro. So thank you very much for the support. We're able to transfer our skills to the local Lao partners. And so through this project, we implemented sample collection in live markets. We implemented novel cutting-edge techniques, efficient pipeline for generating these full length genome sequences. This is unique in Lao PDR, and we are committed to further doing this work. So thank you very much. And yes, thank you very much to the team. Thank you very much to our local Lao partners and also our partners at the Institut Pasteur to Cambodia. Thank you.
Thank you, Gary, for showing this interesting evidence on the utility of environmental surveillance and in particular of air sampling for the surveillance of avian influenza viruses in these settings that are high risk of zoonotic spillover. We're going to move forward to our second presentation by Filipe Moreira, who is a bioinformatician and a genomic epidemiologist at the Federal University of Rio de Janeiro. And he will present on a new pipeline, an offline-capable computational framework for decentralized, real-time, and targeted pathogen genomic surveillance. Thank you, Filipe. Welcome.
Well, hello everyone. I realize that my title is a bit large. Well, good afternoon, and thanks Silvia for the introduction. Well, my name is, as she said, my name is Filipe, and I'm very happy to present the work my team and I have been doing for the past 10 months under the Catalytic Grant Fund. And it's essentially a piece of software for performing metagenomic analysis everywhere. So here's a quick overview of what we're going to see today. I'll start by explaining why we decided to build this tool and what are the problems we're trying to solve. And then I will talk a little bit about the design and structure of this application, and you're going to see what our prototype looks like so far. And then we're going to discuss some of the topics, including the guidelines that we used for development, and go through the deliverables that we managed to achieve. So we all know that portable metagenomics is a powerful approach for detecting pathogens, even when we don't know the content of their genomes. This is particularly useful when in resource-limited settings, where infrastructure for large sequencing projects is often lacking. And when I say portable sequencing, I'm referring specifically to nanopore technology and the MinION sequencer, which can be deployed almost anywhere. This capability is essential for timely public health responses, as rapid pathogen detection helps determine which tests to run, how to monitor cases over time, and what interventions to put in place. So our goal was was essentially to develop this tool, which you call RT-Meta. So in terms of application design, we conceptualize this as kind of two pieces of independent software that are interconnected. One is the VarUnit package here on the left. It's a Python module that manages workflows written in SnakeMake for performing tasks such as taxonomic assignment and consensus sequence building, reference genome assembly, really. And on the other side, we have the RT-Meta app per se, which has a front end and a back end service that sort of provides an interface under which people can actually configure and run analysis. And when I say this component of performing things in real time, I mean it quite literally, because as basic calling happens, we can actually classify reads and understand the contents of the metagenome in real time. So this is like our project has been openly developed. It's on GitHub. It's still a work in progress, but we made a lot of advances. We are going to go through some of that. So I'll quickly show you what the prototype looks like. So we have a front-end interface and a menu where people can add some details of their analysis, like what the databases they want to use for taxonomic assignment, some details as the name of samples, the barcodes that were used for running analysis, all things necessary to configure the analysis, really. And this is without using the command line interface in a very easy way. We can launch the analysis. It takes some time to run. It has some UI features, like several visualization modes. And essentially, once the analysis is run, we can actually see the result in real time. We have these chronoplots, which really show the diversity of the metagenome. And users can go and check what are the contents of the sequencing reads while sequencing is proceeding. And we believe this is extremely useful, because otherwise what usually happens is that people have to wait for the sequencing run to be finished, and this provides critical insight in a very short amount of time. One of the things that we also thought of that could be useful is the capability of comparing different samples. So for instance, you can compare one of your samples with a negative control and check whether you have any contamination background. And of course, you can also compare multiple samples in order to understand the composition of your collection of samples in a public health emergency, for instance. And aside from this, very simple, specific visualizations, we also have nice summaries of, for instance, the viral families that were detected, or the specific viruses in each viral family with how many reads they were supported. And general visualizations indicating the number of reads that were assigned to each viral family are also included. So in general, what we try to do is to generate a tool that actually tells you in different ways what is the content of the metagenome. And I think that's enough for now. So in general, right from the beginning, something that we tried to do was to adhere to some of the PHA4GE guidelines. Basically, who works in this project is myself and 2 other developers, and we read the document carefully. And for some of the things that PHA4GE laid out was really simple to adopt, like open development, like these really reflect the best practices, but other things like benchmarking and software testing are more challenging, and we are really working on it right now. So a few things that I quickly realized that, as I mentioned, this is a work in progress. There are still a lot of work to do. Specifically, there are features that we would like to include in this pipeline that were not included yet. This includes a dehosting option for removing, for instance, human genetic material before performing analysis, and the inclusion of alternative taxonomic tools, because so far it's supported by Chrono 2, but I would like to also include Diamond, for instance. Other aspect that I feel like users would benefit from is using this interface for performing tasks such as risk mapping in real time and consensus sequence workflows. On the long run, we also think of including different types of data, for instance, short reads, but this is something more to the future. And of course, this data will not be able to be analyzed in real time, but this is something that we have been thinking of. So here are some of our deliverables. We have this prototype application. It's alive, it exists. And we have some basic tutorials and examples to run. And we are currently working to perform different aspects of validation and benchmarking. One of the challenges that we faced is the limited support for nanopore sequencing locally. So we are still making partnerships to make that happen. And finally, what we expect to deliver is a collection of, it's a tool, and a collection of examples of real-time use of it, and a report on the critical use of this tool to genomic surveillance in real life. So some of the challenges and lessons. Well, one of the first things that I realized is that professionals with a technological background do not automatically grasp some bioinformatics concepts. They need some time to understand things that are very specific to the field, and communication is really, really important to make this kind of project advance. Other things that I realized is that sometimes you engage very excited in the project, but the adoption of a technique such as metagenomics in public health settings is not automatically great. So we need to build partnerships in this regard. We assembled a partnership with a reference center with PAHO in São Paulo to actually perform some testing, which should happen over the next 2 months. And some of the next steps, we are going to finally implement the features that I mentioned, performing all this benchmark and validation steps, and finally make Artimeta something more broadly available for anyone. Well, sorry, my closing remarks. So in summary, Artimeta aims to make real-time metagenomic surveillance accessible, reliable, and easy to deploy in the field. We are proud of the progress so far, even though there's still a long road ahead. Our vision is that metagenomics and its public health applications should be available for everyone, everywhere, and at any time. Thank you for listening. Oh, by the way, this is my team, some of the cool people that make this possible. Thank you. I'm just in time.
Thank you, Filipe, for introducing this new tool with really useful features. And I hope that during the conference, you had a chance to talk to other bioinformaticians about it and maybe find additional users to test it that may have metagenomics data and that may be interested in sequencing in the field. All right, so we are going to move on to the last speaker, which is, unfortunately, wasn't able to secure a visa to be here in person. So we're going to hear about his project through a video recording. Rodrigue Baltasar Ayagriwe is an assistant professor at the Evangelical University in Africa in the Democratic Republic of Congo.
Ladies and gentlemen, all protocol observed. I'm Rodrigue Ayagriwe from UEA, Bukavu, DR Congo. So it is a pleasure to introduce to you our project on generating genomic surveillance data of pathogens in the DR Congo by extending the mini lab with a nanopore minion sequencer. So the DRC was considered as a blind area regarding genomic pathogen surveillance, and yet from the early initiated mini lab in Panzi Hospital, we have been able to isolate various pathogens reported to be resistant to antibiotics. So in this project, the aim was to extend that mini lab with a minion sequencer to train scientists and to generate results that could help to genomic surveillance and then sustain that project after IPSN grant for continuing of pathogen isolation sequence and analyzing. So far, we have been able to train 3 scientists to perform and analyze the minion sequencing data. As you can see on the photo here left is our training in Brussels at ULB with our partners, as well as the right photo indicating the initiation bioinformatic analysis. We have also been able to install and run the minion sequencer in the Panzi Hospital mini lab. As you can see, the trained scientist and performing sequencing in the mini lab. So as a result concerning the diversity of isolated bacteria, so far the result has indicated that Salmonella typhi, Salmonella pneumonia, and Escherichia coli were the mostly prevalent isolated bacteria out of the 90 different strains that we have isolated so far. So we tried now to make some association analysis, and as you can see highlighted in that B plot, for example, considering the Klebsiella pneumoniae enterica, as well as Salmonella and Escherichia coli, were the mostly isolated bacteria related to internal medicine as well as the surgery care unit. And we have already identified resistant genes that are associated, for those different bacteria that are isolated, and this is mostly for patients with above 40 years old. So we did some first batch of sequencing 8 samples, and after analysis, we have seen a diversity in terms of sequence type related to the 4 different species that have been sequenced so far. So we tried to look at their carried resistant genes and predicted phenotype. And here we highlight only the genes that are responsible to the commonly used antibiotics in our Panzi General Hospital. And as you can see, the Klebsiella pathogen carry genes that are responsible for carbapenem, cephalosporins, and fluoroquinolone antibiotics. Coming to Salmonella typhi, we did the same exercise with sequence type 1, and so far, regarding the commonly used ciprofloxacin antibiotics, we found that Salmonella typhi sequence type 1 carry already the gene, the resistant gene associated to that antibiotics. The same observation has been made for Escherichia coli, where different antibiotics were concerned, and the strains carried a lot of resistant genes associated with the commonly used antibiotics. And as you can see, we have already some third generation antibiotics for which there is resistance. We tried as well to make some phylogenetic trees, and of course, our strains of Staphylococcus were clustered together with the previously isolated strains in DRC and Uganda. And this is normal given the traffic that exists between the two countries. This contrary has been observed with the Staphylococcus aureus sequence type 15, which clustered together with the one isolated in Mozambique. For Salmonella typhi, we tried to analyze our data with the African countries, and the cluster shows that our strains were more closely with the one isolated in Rwanda, which is the closest country with high traffic between the two countries. For Klebsiella pneumoniae, we tried as well to compare what we got as the sequence type with other African countries. And if we zoom in, it was possible to show that our strains are closer to the one that have been already reported in South Africa, in Malawi, as well as in Nigeria. Unfortunately, we couldn't build the trees from strains isolated, sequenced for Escherichia coli, even if we got two different sequence type. However, after blasting, we found that our sequence type observed in this project have already been mentioned elsewhere around the world. So from this project, we have been able to generate results that has impacted the clinical prescription of and the patient outcome, the implementation of sequencing modules within the mini lab, and that opens a prospect for epidemiological surveillance, and it was possible to sequence with the mini lab and unable to consider it as an additional tool for monitoring high contagious diseases. As a take-home message, we have now high diversity of bacteria strains circulating in the region with high prevalence of antibiotic resistance, both at phenotype and genetic profiling. And this highlights a very huge need for One Health antimicrobial resistance surveillance in the region. So let me acknowledge a lot the donor who funded this project, as well as our partners, Utrecht University and the Université Libre de Bruxelles. And thank you for your kind attention. Thanks a lot.
Unfortunately, this is the second year that Rodrigue was unable to be attending the Global Partners Forum in person. I hope that maybe next third time is the charm. So we're going to move to the Q&A, and obviously we're not able to include him in the Q&A, so I invite the two speakers to join me in the stage and take a seat. And in the meantime, I really want to highlight the incredible work that Rodrigue and collaborators have been doing to implement sequencing and genomics and deliver this project in an area that is affected by armed conflict. And this meant from overcoming challenges with procurement to capacity building. And now we had a first look at this data that was literally generated 1 or 2 weeks ago. So over the final months of the project, we will see a lot more data from bacterial pathogens, drug-resistant bacterial pathogens coming out, and they'll be refining their analysis. So stay tuned for the final updates. Okay, so we'll move on now to the Q&A. Are there any questions on the audience for Gary and Filipe? I see a couple of questions, 3 questions. Okay, I'll start here. If you could please introduce yourself and your affiliation, and then who you're directing the question to. Thank you.
Thank you. Sorry, I think I barged in. Hi, I'm Nicola Holden from SRUC in the UK. I've got a question, first of all, for Gary, if I may. So you explained that you did your sampling in Laos in the live bird markets. Were there any other animals that were used for human consumption also present in those markets? And would you be interested in looking at them too, or?
So we sampled 2 live bird markets, so one in Vientiane and one in Luang Prabang. And so at these live bird markets, so in addition to birds, occasionally there may be other animals. We noticed that there were rabbits, sometimes civet cats, but this is not, I would say, consistent. So we don't notice them every time we go. So really what's there are live birds. We didn't notice any other live animals such as pigs or anything like that. They just sold us pork, so there's already been slaughtered beef as well. So these I think are pretty much just live bird markets.
There was another question, the gentleman with the
Yes,
laptop.
thank you so much for the presentation. But my name is Dr. Idriss Adja. I'm from Institut Pasteur of Dakar in Senegal. I have a question for Romero. I think I have the opportunity to use your tool, Viral Unity, when you develop it at the first stage. And then it is a quite easy to use tool, and then it is a big game changer that you put the front end part in order to be able to run without running any line of code. And the question is, I know that I use it for Illumina data. Is the tool be able also to analyze nanopore sequencing data?
Well, thanks for the question, and thanks for saying this. Actually, it does support nanopore data, but the variant calling pipeline is not great yet. I would usually recommend the use for nanopore-specific data, especially if you're working with amplicons. I highly recommend the Arctic pipeline. And I myself have been thinking of actually plugging in Arctic into this workflow, but that feels like a lot of work. Right now, I'm really focusing on the front end and trying to make things in real time work faster and more effectively. But yeah, that's something that's on the map.
Yes.
Thanks so much. I really enjoyed both the presentations, all 3 of them. But my question is for Filipe as well, sorry. So I think looking at the kind of user interface, I think that looks like really amazing. And the part I particularly liked was being able to compare across multiple samples. And really, I think that's one of the things with metagenomic data, right? Like there's so much information, how to actually tease it apart what's of relevance. So part of my question, I'm sorry, it's like multiple little questions. Part of my question is there a way of filtering those kind of comparisons to try and really, like say if it's not something that's particularly prevalent in your metagenomic sample, is there a way of sort of filtering down? Because when we've done it, we've noticed that sometimes if you're interested, say, in a virus, it's really a small proportion of the reads. So I was wondering if that's something you'd looked at. And then across those samples, how does it scale? So how many samples can you kind of include before it starts to not really work? And then also, have you thought about more complicated virus situations, like for segmentation, segmented genomes and that sort of thing? Sorry, there's lots of them.
Okay, I might not remember all questions. I will try to remember in order. So essentially, no, we have not thought of ways of filtering the results, but that could be a feature. One of the things that Artimata is really something not that community-driven so far, but that's the vision long term. So yeah, we should definitely consider this. And one aspect that I was even discussing with some people is that like the possibility of including filters to remove contaminants that I found in the negative control. I feel that for metagenomics that's particularly important. Well, sorry, I said I wouldn't remember. How does, well, yeah, I mean, in my experience, especially running metagenomic sequencing with the MinION, I don't think it's a really good idea to multiplex a lot of samples. Like the most multiplexed sequencing run with these protocols that I have seen is like around 24 samples. And it effectively works, like it runs. The thing is, it depends a lot on the computer running, especially because that was designed to run on a sequencing laptop, which is usually good, but it's not like an HPC. So what happens that if you multiplex a lot, it will run, but it will be a bit slower to update the things in real time.
And then the third part, sorry, was complicated cases like segmented genomes. Are they handled in any particular way?
Oh, yeah. Actually, so far, as the main task is really performing assignment and not genome assembly, which would be another beast, it kind of doesn't matter. What matters is like the k-mers of each segment for each virus is included in the database, so it makes no difference whatsoever. What happens is very often the underlying tool that's used, which is Kraken2, is not very sensitive for new viruses, which represents a particular challenge. This is the underlying reason why we want to include Diamond, because it's more similar search-driven, and it generates more useful statistics to interpret the data.
Thank you. I think there's another question over there. Over here, sorry.
Okay, thank you for your presentation. This is going to be an impactful tool in metagenomics. I would like to know, does this tool contain error correction, for example, for long reads using short reads? And also, does this going to scale to other PHA4GE, for example, because I saw it's majorly for ANC, if I got that wrongly? Okay, thank you.
Thank you. I mean, there is no specific error correction for nanopore reads prior to performing taxonomic assignment, but hopefully nanopore data has been advancing so much and we have better and better base calls. But yeah, we should definitely think of ways of trying to correct for this in the future. And I must confess that I never thought of including other type of PHA4GE data or any sort of data that's different than nanopore and Illumina. But that's something that should come to the map as well. But the only thing is that as I have no experience whatsoever with this kind of data, for me it's kind of difficult to try to imagine it being used in these settings that I see people using this.
I see someone with a microphone over there. No? Okay, are there any other questions from the audience? All right, so maybe I will take one more question for both speakers, and it's related to some of the funding sustainability discussions that we've been having yesterday and today. And of course, this, the Catalytic Grant Fund, it says in the name, it's catalytic, so it's, these are projects that are running for a little bit over a year. And so I would like to know, from your perspective, how you're planning to continue with these projects and how, what kind of sustainability plan you have going forward.
Would you like to go first? Yeah, sure. Okay, so I'll go first. Through this project, this project was actually very, very important for Laos because before that, we didn't have any full-length genome sequencing capacity in the country. Before, a lot of these samples were sent overseas for full-length genome sequencing, and then the turnaround time was at least 6 months. Now it's much shorter, which is nice. And of course, so how do we continue on with this capacity gain? So of course, we want to continue using these techniques for surveillance. Not just necessarily AIV, but also other pathogens as well. And so in my group, we also have other projects, so with emerging viruses and so on and so forth, where we're applying the skills that we learn to sequence some of these other emerging pathogens that may become public health problems. That's one way of doing it. And then, of course, if we were to expand our capacity, in flu, we also want to collaborate with the human infection side, because through this project, this is primarily live-birth market, so we work with the animal people. We also wanted to expand to the human side, so maybe through ELISARI sites, looking to see whether we can have samples from people with severe respiratory symptoms. Maybe they may have bird flu infection. We may be able to try to sequence those viruses as well, which may help guide maybe clinical decisions and so on and so forth.
Thank you. And Filipe?
Well, a lot of this conference, the word sustainability kept emerging, and in fact, we were really discussing this in the PHA4GE Pipe and Viz group yesterday. Well, I think in terms of sustainability, I feel like we should be moving to a model where we have less pipelines competing and more pipelines driven by communities, which means that if you would like a tool, if you use a tool and would you like to contribute, and you can apply for grants to add new functionalities and to keep things running in a way that doesn't depend of a single person. I feel like that would be a major step. And I also to happen to work in an organization dedicated to epidemic preparedness, and we do provide some funding for this kind of work, and I feel like this should be important. And I would highly encourage other funders to also look at that in the way that we need these tools to keep working beyond research-driven settings. We need to bring genomics into surveillance, and to do that, we need to use other indicators and publications to effectively assess the value of these tools.
Thank you. And what I can add from having discussed this with Rodrigue before is that they've been applying for other grants to continue with the work, but it is clear that obtaining sustainable funding from government sources continues to be a problem across these projects. So I'd like to close the session for today with thanking all the grantees that have made it both here on the stage and at the poster sessions and presenting their projects. And just to say, it's an absolute honor to work and support all of you. It's been really one of the greatest experiences I've had in my career, and I continue, I look forward to doing that again.